So today I'm going to talk about how muscle fibers are different than other cells. So what I did is I actually redid the cell, and you can see it here. You can see the nucleus, the endoplasmic reticulum, smooth and rough, mitochondria, Golgi, and the cytoskeleton. And I am going to go into how this cell is modified. Now, before I do, I just want to let you guys know about the how skeleton is made. And you always start with the cell. You end up in the nucleus with the DNA, which gets copied into mRNA. It will leave through a protein channel called a nuclear pore. And if you're making proteins that are going to be outside the cell, it will go to the rough endoplasmic reticulum, where it's going to be made in a membrane, pretty much. And then it'll go to the Golgi, and then the Golgi will actually send it out of the cell. And so you'd end up having protein structures around the cell. With a muscle cell, a myocyte, we're actually going to get a little bit different. Now, I'm not going to talk about all the types of muscles. What I'm going to talk about is striated skeletal muscle. Striated means striped, skeletal means it's with the skeletal system, and muscle means it's a contractile tissue. And one of the first things that I'm going to tell you that happens is we are going to deal with the cytoskeleton. Now, usually what happens is you do have some sugar molecule come in here. Enzyme breaks the sugar, making it pyruvate. The pyruvate will be modified, go to the mitochondria. Mitochondria will use oxygen to really break the sugar molecule that went in, the pyruvate technically, not the sugar molecule. And it will break it down into carbon dioxide and water, which will leave the mitochondria, get into the cytoplasm, which is a fluid in here, and then leave the cell. The mitochondria will be important for some things. And what you're doing is you're breaking down sugar, and when you break down sugar, you get energy. And you use that energy to get adenosine diphosphate, a adenosine with two phosphates attached to it, and a inorganic phosphate, and you bind them together to get adenosine triphosphate, three phosphates. And you will use that ATP, adenosine triphosphate, for energy. Now, what we are going to do is we're going to modify the cell. So what I'm going to do first is remove these proteins, which I'm going to use later. And what you have is you actually start modifying the cell by taking going to move the mitochondria for now, start making these thinner filaments called actin, and you have thick filaments called myosin. I'm going to do some of the thin filaments here, and I'm going to actually put a couple thin filaments over here, which they are going to eventually bind to the cell next to it. I'm moving the mitochondria, the nucleus, the rough endoplasmic reticulum. We don't need it right now. And what we're going to do is we're going to make thin filaments and alternate it with thick filaments. So we have thin filaments here, and between it we'll have a thick filament called a myosin protein. And it is part of the cytoskeleton's protein. And so we would have a lot of these, and I'm going to break this one so you can actually have more. And so what we're doing is we're modifying the cell to make it have thin and thick filaments in alternating arrangements. And so this is actually a thin filament here, thick filaments here. This is made from a protein called actin. These thick filaments are made from a protein called myosin. And usually each myosin will have two heads and two tails. And these will actually intertwine to make the thick filaments. In the head, you have an ATPase. ACE is an enzyme, and ATPase will break this enzyme and use that to cause a contraction. Now, as you have thin and thick filaments, we actually do name a lot of these structures. And I will tell you the names of these structures as we are doing it. So, what we have is, right now, we're actually seeing that we have this arrangement of molecules, actin, myosin. And you do have other proteins which will actually connect here, which will give you a bigger, pretty much a bunch of different things that you can see. So you can actually see an area in the middle here, an area in the middle here. Now, the area where all you have is a myosin, you can see here, there's thin filaments on the side. The areas where you just have myosin is called the M line. The area where you have myosin and actin is the A band, and where you just have the actin here is the eye band. You do have other bands that you deal with a lot in physiology. And what ends up happening is this. Like I said, the ATP is broken, and the myosin will pull actin close this way, and this actin this way. And so eventually, what you do is you shrink the cell. But we're not done. There's actually a lot of other things that are going to happen with this structure. One of the things that we're going to do is we're going to take the smooth endoplasmic reticulum and modify it. The way we're going to do it is it's going to actually come into the smooth muscle. And when it does it, we change the name of the endoplasmic reticulum to the sarcoplasmic reticulum. The sarcoplasm means flesh. And on these things, you have these little tubules that come off of it, called T tubules. I'll make it look like a little T, that go deep into the myofibrils. All these fibers together are going to act, each individual is myofibrils. Now remember, these are pretty much cytoskeleton proteins. And as you do have a nucleus and you do have ribosomes, you still have some rough endoplasmic reticulum as well as a smooth endoplasmic reticulum. What you're going to do is you're going to be able to build more of these, and so you can actually expand the way the muscle actually works. And so the cell has everything that a cell would normally have, and depending on how active it is, it may have multiple mitochondria, or it can actually lose mitochondria. And a lot of times we'll talk about type 1 fibers, type 2A, type 2B, and type 2X. Type 1 fibers tend to be postural muscles, and they tend not to necessarily contract too quick. They have a lot of blood going to it, and so they have a lot of oxygen, and so the mitochondria can make a lot of ATP. These muscles may not move fast, but they can maintain a contraction and don't necessarily get tired or fatigued too much. The more mitochondria, the more oxygen demands it needs, and the slower it tends to move. As you actually get faster and faster muscles, these muscles just have to get rapid contractions. And so what we have is we have pretty much fast contractions which are not sustained. So if you think of your back, as you stand, your muscles are constantly contracting. If you think of your arms, you're usually doing a quick motion and relaxing until you have thin. The type 1 and type 2. Type 1 tend to be more postural, type 2 tend to be active. Now in physiology, you will actually break it apart into type 2A, B, and X. But for now, we'll deal with this. The other modification that ends up there's a lot of modifications that ends up happening. One of them is you do have transmembrane proteins, which are channels which allow sodium, potassium to move in, sodium, potassium, ATPase. And you have receptors, which I made one over here. And so neurotransmitters can bind to this to change the charge of the membrane, which will actually work on the sarcoplasmic reticulum, the modified smooth endoplasmic reticulum, which has calcium. Now, I didn't draw it in here, but you do have a regulatory protein bound to the actin called troponin. And when the calcium from the sarcoplasmic reticulum is released, it binds to the troponin, moves it out of the way, and so actin and myosin can then interact. And so you can end up having a contraction. The other modification that happens is this cell fuses with a bunch of others, and so you end up having a thin fiber, multiple nucleated fiber, which pretty much goes the whole length of the muscle. The reason is that way you can get a contraction which is smooth. And there are a lot of other things I'm going to tell you about in muscles, and that is this. So you have the muscle fibers, and the muscle fibers still have the cell membrane around it. And I'm actually going to make it smaller so you can see what I'm talking about. So this cell membrane would actually go all the way around it. You'd still have the nucleus pushed to, and so you end up having a muscle fascicle. Now you'd have a bunch of muscle fascicles, and so what I would do is I'll take a couple of these muscle fibers and deal with how we would actually end up packaging them. So we'd have a bunch of muscle fascicles, these entire cell, and we'd actually, like I said, we'd have a membrane around here, and that membrane is called the perimysium. The endomysium. So these would actually be wrapped together, and you'd have the endomysium. Endo means inside, mycium, myo means muscle. So the myosin, which when I put it here was here, would be the muscle protein. When this entire thing is bound together, you would actually have this with the phospholipid bilayer around it, which would be the endomysium. Now, you would end up having a bunch of these together with endomysium, and then around it, you would have something called a perimysium. The perimysium would pretty much take the muscle fibers bound together, making something called the fascicle, holding it together. And between the different perimysiums, which would be wrapped together, you would end up having blood vessels and nerves. The nerves, again, to give you the message to contract. Blood vessels will, again, feed this. And around a bunch of fibers which are wrapped with the perimysium, you then have the epimysium around that. And the epimysium pretty much starts putting all these things, all these different fibers together, the fascicles together. And what you end up with is, you end up with the muscle wrapped in loosely, pretty much connective tissue. And then around that, you have another thing called an epimysium. So you have endomysium, which is the inside holding the fascicles together. You have the perimysiums holding a bunch of fascicles together, and then you have the epimysium on top of that. So endo inside, peri around, epi above. Now around, connective to the epimysium, you have tendons, which are dense regular connective tissue, which are bound to bones. Now this is actually just a short video because all I wanted to talk about is how the modification of the cell and how you have different structures that are the same as in any other cells. And so if you can understand one cell, you can see how the modification works. You can see how the sarcoplasmic reticulum is just a smooth endoplasmic reticulum modified to take in calcium, usually using ATP, so calcium ATPase, to pump calcium into the sarcoplasmic reticulum. And when you need it, when you have a neurotransmitter bind and you want to actually get a contraction, it releases the calcium. The calcium will bind to the troponin that's bound to the actin. And then when it's moved, the myosin can act with the actin, can break ATP and start causing a contraction. Now to get the contraction to relax, you need to remove the calcium first and bring it back to the sarcoplasmic reticulum. You do that by actively pumping it back in. And so to get something to relax, it's more of an active process. That's why if someone goes to the gym and works out too much, they end up maintaining that level of increased contraction, which is due to the fact that you haven't returned the calcium back to the sarcoplasmic reticulum. As you notice, we are actually modifying the cell, and so I will always go back to the cell so you can see what's going on. I hope you enjoyed this, and you'll have a nice day.